Very good morning to my dear students of class 11 science. Welcome to biology class. We are dealing with the chapter anatomy of flowering plants. So we'll continue with the same chapter. So today we are going to learn about the secondary growth in stems. The secondary growth in roots have been removed from our new read syllabus. So we are only going to deal with the secondary growth in stem. Clear? So first of all, what the secondary growth means? So when we look at the growth of the plant, then only the stem and the root of the plant keeps on growing. Okay, so the growth of the root and the stems from the apical meristem part of the plant is called the primary growth, whereas the increase in the thickness of the plant, okay, due to the activity of the cork cambium and the vascular cambium, is called the secondary growth. So one more telling you that whenever there will be increase in the root and the stems length, then that is called primary growth. But due to the activity of the cork cambium and the vascular cambium, whenever there will be increase in the thickness of the plant, that is called secondary growth. Clear? So here the two cambium are involved, that is the cork cambium and the vascular cambium. So these two cambium are involved in the secondary growth of the stem. So therefore these two cambium, the cork cambium and the vascular cambium, they are collectively also known as lateral meristem. So apical meristems are involved in the primary growth of the stem and the root, whereas the lateral meristems are involved in the secondary growth in the stem and the roots. Clear? Okay. Now the secondary growth in the stems can be discussed under the different headings. Okay, first, like activity of the cambium we are going to discuss, but listen very carefully that the secondary growth in the stems is mainly due to the activity of the vascular cambium and the cork cambium. Clear? So if you remember that we have learned in case of dicot stems that in between the xylem and the phloem we will find the cambium. If you remember that we have learned in the previous classes also that in case of dicot stems, in between the xylem and the phloem we will see the presence of cambium. Clear? And those are called intrafascicular cambium. Listen very carefully. Okay, so just look at this figure. If you look at this figure and just try to recall it back with the topic, the dicot stems, you remember that over here we have got the xylem, we have got the phloem, and in between the xylem and the phloem we have got the cambium. So it is called intrafascicular cambium. Clear? Now during the secondary growth of the stem, clear? Listen carefully that during the secondary growth of the stem, the cells of the medullary ray, okay, that runs through the vascular bundles, okay, the cells of the medullary ray, it lines with the intrafascicular cambium and develop the meristematic activity and form the strip of cambium called the interfascicular cambium. Now the intra and the interfascicular cambium, they will form a complete ring which is called the vascular cambial ring. Now which one is intrafascicular cambium, which one is interfascicular cambium, and what do you mean by vascular cambial ring, we are going to look over here. Okay, so if you look over here, we will just see the structure. Clear? Okay, so look over here. So here you can see the presence of cambium in between the xylem and the phloem. Clear? Now if you look over here, we have got the cambium which is called the intrafascicular cambium, because here you can see the presence of primary phloem and the primary xylem. So this one is the xylem, this one is the phloem. Okay, so this is the phloem and this is the xylem. Now in between the primary xylem and the primary phloem you can see the presence of cambium. This is called intrafascicular cambium. Alright, now during the secondary growth of the stem, this particular intrafascicular cambium, or let's say along with the medullary ray, it will form the interfascicular cambium. So this one is the interfascicular cambium. Now the intra and the interfascicular cambium, they will form a ring. Can you see this ring-like structure? So this ring-like structure will be formed. Now, therefore, this ring-like structure which is being formed is known as vascular cambial ring. Okay, so the activity of the cambial ring gives rise to the secondary growth of the stem. Clear this one? So here you can see one more time the intrafascicular cambium, then we have got the interfascicular cambium. Now the intra and the interfascicular cambium, they will unite to form the vascular cambial ring, and the activity of the vascular cambial ring will give rise to the secondary growth of the stem. Okay, now let's talk about the formation of secondary tissues. Okay, number 2. So that's now we have learned about the activity of the cambium. So remember, interfascicular cambium and the vascular cambial ring. Clear? Now number 2, we are going to learn about the formation of secondary tissues. Okay, so as we have learned that the activity of the vascular cambial ring will give rise to the secondary growth of the stem. Okay, so therefore the cambial ring becomes active as a whole, and when this cambial ring becomes active, it starts cutting off new cells. It starts producing new cells. So the cells which has been cut off will be differentiated into phloem on the outer side, which is called the secondary phloem, and the cells which will be cut off on the inner sides will be modified to form the secondary xylem. So one more time saying that once the vascular cambial ring becomes activated, it starts cutting off the new cells. That means it starts producing the new cells. Now the cells which will be differentiated towards the outer sides, okay, that is towards the phloem, will form the secondary phloem, and the cells which will be cut off toward the inner sides, okay, toward the xylem, will form the secondary xylem. So outer side we have got the phloem, so it will form the secondary phloem. Inner sides we have got the xylem, it will form the secondary xylem. Now secondary phloem is mainly composed of sheath tubes, companion cells, phloem fibers and phloem parenchyma, whereas secondary xylems is mainly composed of xylem vessels, xylem tracheids, xylem fibers and xylem parenchyma. Clear? So here we have learned about the formation of the secondary phloem and the secondary xylem. Okay, so activity of the cambial ring will be more toward the inner sides than that of the outer sides. Listen very carefully. So activity of the cambial ring is more toward the secondary xylem, toward the xylem sides compared to the phloem sides. Clear? As a result, there will be a rapid bulk next towards the xylem sides. So due to the increasing pressure, primary phloem is completely crushed and is represented by small patches, and the primary xylem is pushed towards the pith, that central part, okay, and remain more or less intact. Why? Because the activity is more toward the inner sides compared to the outer sides, and as a result the xylem will increase more rapidly compared to the phloem. And when the xylem starts rapidly producing, when it increases rapidly, it will give or it will exert a pressure toward the phloem, okay, and as a result the phloem will it will receive enormous pressure and it will be crushed out completely and will remain in the form with small patches. Okay, now let's talk about number 3, the formation of secondary medullary ray. Okay, so certain cells of the cambium, they form some narrow bands. Clear? That means the medullary ray of the cambium, they form a certain band, okay, that pass through the secondary xylem and the secondary phloem. Now these narrow bands that pass through the secondary xylem and the secondary phloem, now they will be known as the secondary medullary ray. Okay, now what is the function of secondary medullary ray? It provides the radial conduction of food from the phloem and the water and salts by the help of the xylem. Next one, what do you mean by spring hood and the autumn hood? So vascular cambium is said that it remains active throughout the life of a plant, but its activity is not uniform in those plants which grow in regions where favorable climatic conditions alternate regularly with unfavorable climatic conditions. Though it is activated throughout, but you can say that its activity is mainly under the influence of climatic conditions. Okay, so in temperate climatic conditions, the cambium becomes more active in spring. So in temperate climatic conditions, this cambium becomes more active in spring and it forms a greater number of vessels, okay, and forms narrow and smaller vessels. So the hood which are formed in the spring is called the spring hood or the early hood, and which are formed in the dry summer or the cold winter are known as the autumn hoot or the late hoot. So remember, what does the spring hoot and the autumn hoot means? So we said that in temperate climatic conditions the cambium becomes active in the spring. Okay, so therefore the hoot which are formed in the spring are called the spring hoot or the early hoot, and those hoot which are formed on the dry summer or in the cold winter, they are called the autumn hoot or the late hoot. Alright, okay, now let's talk about the next one, the cork cambium or the formation of peridome. So we'll go with the figure number 6.37 to look at the structure of the cork cambium or the formation of peridome. Okay, so the marked increase in the diameter of the stem due to the addition of cell layered by the secondary growth exert great pressure on the outer tissue, because the activity of the cambium will be more towards the inner side compared to the outer sides. Okay, so as a result, the outer sides will exert a greater pressure. So this results in the rupture of the cortex and the epidermis. So since the outer tissues will exert a great pressure compared to the inner sides, so therefore the bark will rupture. Okay, so the bark, or let's say the cortex and the epidermis, will rupture, and as a result, due to this one, the outermost layer of the cortex, it becomes meristematic tissues and it begins to divide, and this is called the cork cambium or the phylogeny simply. So due to the secondary growth of the stems, and since the activity of the cambium will be more toward the inner side compared to the outer sides, so the cortex and the epidermis which are present on the outer sides will rupture. So you can see here also the epidermis part, okay, the outer part, it will rupture. The cortex also ruptures, the epidermis will also rupture, it becomes meristematic and begins to divide, and this is called the cork cambium or the phylogeny. Okay, now the cork cambium forms the secondary tissue, but its activity is more on the outer sides than that on the inner sides in this thing. Okay, so cork cambium activity will be more toward the outer sides compared to the inner sides. Now the cells which are formed on the outer side, it forms the cork or the phylum, and those on the inner side, it forms the secondary cortex or the phylodome. So you can see over here, so as we are telling that the cork cambium activity will be more toward the outer side compared to the inner side, so therefore the cells which will be formed on the outer side, they will form the cork or the phylum, phylem phylum, and the cells which will be formed on the inner sides will be known as secondary cortex or the phylodome. Okay, and this secondary cortex is mainly formed of living parenchyma cells. Now here the three tissues which are formed. First one, we will see the cork cambium. Okay, the first tissue which is being formed is the phylogeny or the cork cambium. So here you can structure this one, the cork cambium or the phylogeny, this structure. Okay, second one, you can see phylum or the cork. So you can see over here the cork, which is also called the phylum, phylem phylum, and the third one is called the phylodome, the secondary cortex. Here you can see the phylodome, this portion, the phylodome. Okay, so this cork cambium, the cork or the phylum and the phylodome, they are together known as peridome. Okay, and these are mainly formed by the activity of cork cambium. So cork are mainly new cells which are produced by the phylogeny on the outer sides, because its activity is more toward the outer sides, but they soon become dead due to deposition of suberin. Okay, and these cells will mainly form the cork or the phylum of the plant. Next one, we'll talk about the bark, which include both the living and the non-living tissues lying outside the vascular cambium of dicot stems. Okay, now those bark which are formed in the early spring season are called the early or the soft bark, and those bark which are formed on the late spring, that mainly on the, let's say, on the winter time, are mainly called the late bark or the hard bark. Okay, next one, lenticels, the opening which are present on the older stems. Okay, so lenticels are the pores which are present in the bark of the stem, and through these lenticels there will be exchange of gases. We have even learning class also about the lenticels, okay, which are the small opening which are present on the bark of the stem, and this lenticels will also help in the exchange of gases. Clear? Okay, now next one we will learn about the significance of secondary growth in stems. Now the importance of secondary growth in stems is to increase the girth of the plant or the thickness of the plant, so that it can provide the mechanical support and mechanical support of the plant. We can say it replaces the old non-functioning conducting tissue and will help in the conduction of water and solute to the distant places in the plant. And number 3, the cork or the bark protect the inner tissue from the injury, heat and the cold. Clear? So this is the significance of secondary growth in the stem. Now the next topic that is in our syllabus is the difference between the hardwood and the sapwood. Clear? So let's see what the difference between the hardwood and the sapwood. But before that, just for your information, what does softwood and hardwood means? Okay, so those wood which are produced by the gymnosperm, they are called softwood, and therefore the wood of the dicotyledonous trees are called the hardwood or the porous wood. So the difference between the softwood and the hardwood. So the wood which are produced by the gymnosperm are called the softwood, and those which are produced by the dicotyledonous trees are called the hardwood. Now what is the difference between hardwood, H-E-A-R-T, hardwood, and the sapwood? We said that in the old tree, secondary wood is differentiated into a centrally situated darker and a harder wood. So mainly in older tree, we can see the zone of differentiation. Alright, we can just differentiate the zone. So on the inner side we have got the darker and the harder wood, which is called the hardwood, and on the outer side we have got the light colored zone or the light colored wood, which is called the sapwood or the albumen. So let's see the difference. Okay, so table number 6.27, hardwood, it forms the central wood. So mainly in the older stem, in the central part we have got the hardwood, and on the outer side we have got the sapwood. Now hardwood is dark colored, whereas sapwood is light colored. Hardwood is heavier, whereas sapwood is lighter. And hardwood living cells are absent, but in sapwood it contains the living cells. Okay. In hardwood, it is the dead and non-functional part of the secondary xylem, whereas sapwood, it is the functional part of the secondary xylem or the wood. Hardwood does not conduct water and minerals, whereas sapwood, it helps in the conduction of water and minerals. Hardwood, the tracheal elements are blocked by the thylaces and other substances, whereas in sapwood, the tracheal elements are not blocked by the thylaces and other depositions. So here we have finished the chapter Anatomy of Flowering Plants. In the next class, I will begin with the chapter Cell Division and Cell Cycle. So this much for today. See you in next class. Thank you.